Okay, so we'll go ahead and get started here. We have just a few announcements for those of you perhaps tuning in a little later. Since we won't be here, it originally did have it on the schedule that your papers were due tomorrow and your vocab, and you had a vocabulary evaluation. I think what's best for us, for those of you who are here, you can already see that that has been moved to the right one day. So have your papers due on Thursday, and we'll have your vocabulary evaluation on Thursday, as well as look at your vocab terms. There were some questions in the prior section on what some of these definitions possibly were. So we went through and just summarized these to what we see here. A nucleotide structure, okay, what is DNA helicase, what is DNA polymerase, as well as RNA polymerase. So anything beyond that, I would presume that you possibly have wrote out already, or still in the process of writing out. Which, I think we have a good start on it. You'll probably be just fine. Okay? So, if you do have them done, what might be a coding sequence? Okay, this is where you have to look at your paper, because I probably wouldn't know it yet either. That remains in the final messenger RNA or mRNA. So coding sequences that remain in the final mRNA. Is that possibly an intron or an exon? An intron or an exon? Well, we can come back to that at a later time. So it's at this time, we'll go ahead and hit pause for those of you tuning in later, and then we will get rolling here again in just a few minutes. Okay, then for the rest of you here, why don't you go ahead and open up to page 336. Some of this we have read before, so 336. So some other, let's say, topics that we had discussed last hour is, okay, if you were to look at this, for instance. Now, this is a very vague description on what it is we're looking at. Okay, at a very basic definition, what does that have to do with those four letters, those shapes? And that is one of the questions on your paper. And I think you can see the answers that on page 329, maybe. Otherwise, if you looked at page 329, yes. So what would you guess we're referring to here? And if we don't know, you can see that on page 329. Well, yes, those are nucleotide structures, but some are purines, the other ones are pyrimidines. So which one's which? That's a purine, this is a pyrimidine, which means that if this is a purine, this must be adenine and guanine, then this is, it's not that one is over the other. So I'll put cytosine and thymine, because that's both of those, okay? Because What does adenine bond to now? And I know you know this because you did an activity on it. Does Okay, so then adenine bonds to thymine, cytosine bonds to guanine. Now, that's if we're talking about DNA. How is DNA structurally different than RNA? Yep, uracil. Then if we were to see this, or just this, which one of those could you presume is DNA and which one is RNA? DNA is on the right? That's incorrect. Oh, RNA is on the right. Okay, now, that's correct, but what makes that correct? In other words, this might be a clue. Yeah, so DNA's got 2 strands where RNA only has 1. Okay? Because that's going to be important when we talk about protein synthesis. Because from chapter, 7, okay, where we talked about different types of organelles, we talked about ribosomes. Those are found on the endoplasmic reticulum. Because what is happening at that ribosome? It's what this whole process is about. It says it on Wednesday and it says it on Friday. Protein synthesis. So what do you suppose is made at the ribosome? Proteins. That's right. Okay. So again, we had just gone over what a nucleotide structure is. DNA helicase, really what that is doing, and you probably have this wrote down somewhere. We talked about this replication fork. You have this circle, it's an enzyme, and what were these dots in here? No. Nope. What type of bonds? Are you sure? Well, what does it say right here? Yeah, that's right. Hydrogen bond. Well, and if we can't see, we can certainly move closer. All right. Well, if it's that big of a problem, I would say move closer. You have that opportunity, okay? So if this is helicase, well, what is it that follows right behind it and seals that strand back together? It has to be another enzyme. And what enzyme would that be? That's in plants. Yeah. So what is it that would be sealing this back together? If this is breaking the hydrogen bonds, what's putting it back together?
Oh, no, that's the DNA polymerase, right?
Yeah, the polymerase, DNA polymerase. That's right. Okay. And then finally, RNA polymerase, that's what we just described, begins the process of transcription. Okay. So, page 336, where it says RNA, about halfway down. Okay. RNA is a nucleic acid that is similar to DNA. Okay. But some differences between DNA and RNA is DNA is double-stranded. RNA is single-stranded. It also has uracil that RNA has. Okay. However, RNA contains the sugar ribose. The base uracil replaces thymine and usually is single-stranded. Now it says usually, but for our purposes, it is single-stranded. 3 major types of RNA are found in living cells. mRNA, and that's how you would typically say it, look at it, just the letter M, and then RNA, but it's called messenger RNA. Molecules are long strands of RNA nucleotides that are formed complementary to one strand of DNA. Okay? They travel from the nucleus to the ribosome to direct the synthesis of specific proteins. That's why this is called protein synthesis. You're making these proteins. So, another aspect is if that message gets to the ribosome because of messenger RNA, well then, we all right over there? Okay. So then, can DNA leave the nucleus? Can it? No, DNA cannot leave the nucleus because that's how we know how to assemble these proteins, because the message has to get from the DNA that can't leave the nucleus to make its way to the ribosome to start assembling these amino acids. Okay. And then it talks about ribosomal RNA. We're not too concerned about that. Okay. The third type of RNA is transfer RNA, are smaller segments of RNA nucleotides that transport amino acids to the ribosome. And Table 2 compares the structures and functions of the three types of RNA. We're mainly concerned with transfer RNA and messenger RNA. We're not too concerned about ribosomal RNA. So then we had said in the Middle Ages, who was the type of individual that could read and write? What did we call them? Well, they're literate, yes. You're close. Scribes. So a scribe is one who could read. Okay? And if they could read, they could probably write. So we take that same terminology, especially the idea of reading, okay? Because you see where this comes from. Transcription just means a type of scribe, something that's reading something. So on page 337, the first step of the, they say central dogma, I don't like that, but it says in the book, involves the synthesis of messenger RNA from DNA in a process called transcription. Through transcription, the DNA code is transferred to messenger RNA in the nucleus. The messenger RNA then can take the code into the cytoplasm for protein synthesis. So what's happening is that messenger RNA is reading the code. It's reading the DNA to take to the ribosome. Okay? Follow along with the process of transcription. The DNA is unzipped in the process of in the nucleus, and RNA polymerase, an enzyme that regulates RNA synthesis, binds to a specific section where a messenger RNA will be synthesized. As the DNA strand unwinds, the RNA polymerase initiates messenger RNA synthesis and moves along one of the DNA strands. Okay, so what it's doing is they're showing you that in figure 13 up at the top, is that it is reading the DNA. That green blob you see there, that's RNA polymerase. So what that is doing is that's reading the instructions on the DNA. Okay. And then at the bottom it says uracil is incorporated instead of thymine. Of course, we knew that. And the messenger RNA nucleotide is made. Eventually, the messenger RNA is released, and the RNA polymerase detaches from the DNA. The new messenger RNA then moves out of the nucleus through the nuclear pores into the cytoplasm. So it then has the code or the message to take to the ribosome. Okay? So we're not worried about RNA processing. But on 338, that's something that you'll be using tomorrow as well. And you did that in your activity to come up with specific amino acids. Okay? I should say they're coding for amino acids. Okay. Because what we see here is we're going to skip the first paragraph, skip the code, okay? Skip the first paragraph where it says the reasoning meant, okay? That's where we're at on page 338 now, okay? It's underneath connection to math. Then you go to the next paragraph where it says the reasoning meant that the code was not contained in the 3 base pairs themselves, but most run along a single strand of the DNA. Okay? Experiments during the 1960s demonstrated that the DNA code was indeed a 3 base code. The 3 base code in DNA or messenger RNA is called a codon. Okay? So, if we're talking about transfer RNA, if it's not a codon, what might that be in your vocab terms? What's the opposite of a codon? An anticodon. That's right. So each of the 3 bases of a codon in DNA is transcribed, or you could say, or is read into the messenger RNA code. Okay? So that figure 14 shows a dictionary, and you did that in your activity. Okay? So figure 14 shows a dictionary of the genetic code. Notice that all but 3 codons are specific for amino acids. So there are 3 of them that don't have any amino acids in them. Why is that? Because some of you came up with these and it was still correct. You came up with this where it comes along and it does this. It can't go any further. It's a what type of codon? It's a stop codon. That's right. Okay? So then when you look at that dictionary or table or figure 14, a lot of them are coding for very specific amino acids. We talk about that if you take the biochemistry portion, these structures and these actually have specific structures to them. They just don't mean anything to you right now. Like if you were to ask your older sister, I know what those structures are because we had to draw them in the biochemistry class. And it would be others as well, but they're not here right now. Okay. So the next paragraph, it says translation. Okay? In other words, if transcription is reading a message, well then a lot of you that are in the Spanish class could probably talk to each other in Spanish and then tell me what you said, because I don't know. So then when you take information from one language and say it back in another language, what are you doing? You're translating. What are we about to read about? Translation. So it's where you're taking information from one aspect and changing it to another. You are translating the code now. Okay. Translation. Once the mRNA is synthesized and processed, it moves to the ribosome in eukaryotes. Okay? This means that the messenger RNA must leave the nucleus and enter the cytoplasm. Once in the cytoplasm, maybe I need to retract what I said at parent-teacher conferences. Do we? Oh, okay. I don't know. Depends what your parents said when they talked to you. Oh, okay. Well, maybe to your lab partner then. I don't know.
No, I didn't really hear a lot.
Okay. Once in the cytoplasm, it's called 5', end of the messenger RNA connects to the ribosome. This is where the code is read and translated to make a protein through a process called translation. Okay? So we're going to take a DNA sequence tomorrow and we're going to walk you through all of those different steps. What is RNA polymerase? What is a codon? What is an anticodon? What is the ribosome? What are the nucleotides in there? Okay? once you start hooking those together, if you have a set of amino acids hooked together, what do we call a group of amino acids? What does it say over there? Proteins. So when you start hooking amino acids together, you are forming specific proteins. Okay, so that's all we had discussed in the earlier section. So vocab quiz on Thursday, papers due on Thursday, vocabulary due on Thursday. Here we had some terms for you, and I highly recommend that you really take note of what we'll be doing tomorrow. It'll be on the Science Channel. That's talking about protein synthesis, because that is a lot of what your evaluation is about. And it's one of those opportunities, and that's what it is. It's an opportunity. Don't leave these points behind. Okay? All right, so we will catch up to you next time.